I am. Good morning, Jimmy. Good morning, everybody. Can you hear me?
Yes, thank you, Eric. Please go ahead.
Great. Thank you very much. Hi, everyone. So this is a brief presentation that both Dieter and I will participate in. And really, it's just to summarize a few of the things that are in the news that are changing, that are maybe a little confusing, to try and summarize what the state of play is at the current time. So I don't have control of the slide, so I'll say next slide, please. So we'll briefly cover issues of how the virus has been spreading and the rates of takeoff. And I think this is particularly important when we look at what's going on in sub-Saharan Africa, particularly at the moment. We'll talk about masks. There've been a lot in the news. Some thoughts on therapeutics, and then we'll return to the issue of antibodies that I covered in a previous presentation and just touch base a little bit on that, although the point with that is really that not much has changed. So next slide, please. Right, so I'm going to show you some numbers that relate to two countries. I've chosen these because they illustrate two ends of the spectrum. This is why I've chosen them. The first is in the UK, looking how now that lots of isolates have been collected from patients, work has been underway to look at the genetics of the virus and understand the genetic fingerprint of SARS-CoV-2. And by doing that, knowing, as virologists do, that viruses small mutations that don't necessarily impact on the way they present as an illness, we're able to track the small scale, if we can call it that, evolution of the virus and understand where it was introduced, who it was moved to, and basically track the movement of it through a population. And what's been shown, so these data come from, now this is almost a month old, shows that in the UK, at the beginning, there were around 1,356 introductions, independent introductions of SARS-CoV-2 into the UK. And the authors of this, and I've got at the bottom there the reference, like most of the references associated with COVID, these aren't yet peer-reviewed, but nonetheless. 1,356 separate introductions into the UK over a short period of time. Many of those introductions have become very rare or became extinct. So they created short chains of transmission and then disappeared, and others were more successful, infecting 2, 3, 4 people, and then onwards 2, 3, 4, and then 2, 3, 4, and growing in that sense. The rate of introduction into the UK changed substantially over time, so that at the beginning there was a large number of introductions, and that then fizzled off towards the end of March as movement controls came into place. And in the UK context, this is basically associated with the half-term school break, when people went off on their skiing holidays or doing whatever international business travel as well, of course, and people returning over a short period of time with these viruses. And the other interesting thing is that the finger is not to be pointed at China in terms of where these cases came from, remembering, if you can remember that far back, that already there was an epidemic raging in Italy and other parts of Europe. And so 29% of the introductions into, 34 of the introductions came from Spain, 29 from France, 14 from Italy, and 23 from other countries. So that's just to show, in a country that suffered a very explosive outbreak, which was very difficult to get under control, 1,300 introductions or so. Next slide. The colleagues at the Kemri Wellcome Trust Center in Kilifi have done something a bit similar for Kenya. And they have shown that there's evidence of multiple introductions again of the virus into Kenya, but at the time that this was done, the number of introductions was of the order of 10, perhaps 20 introductions of the virus. And that tree that you see in the image is how that virus then moved with these, as I said, minor mutations to infect more people onwards once it had arrived in the country. And there's evidence of transmission between Nairobi and the coast. So it would have come into Nairobi, but then transmission to the coast prior to the introduction of the movement restrictions. So the virus escaped out of Nairobi before the clampdown happened in Kenya. And what we see here is a picture that will be similar to that in many other countries, that it introduced, spread, evolved, and moved on. But the key thing, next slide please, is that the number of introductions was vastly different to a place like the UK. And yeah, oh yeah, so sorry, I forgot, I need to cover this. The virus spreads into new countries through the movements of people, and that's really the key thing. Once in a country, it still spreads through the movement of people. Lockdowns are designed to prevent that spread between people. And of course, unlocking, as we're seeing, if we can call it that, in many countries, increases that risk because people mix more than they did before that. So a key message, I think, from today's get together is really to take care in this phase of the pandemic. If you're in Kenya, if you're in Europe, wherever you are, that things are unlocking. It's easier to go to church, it's easier to go to the pub, it's easier to go out, but that does create additional risks. And given that this is a human-to-human transmission problem, we need to take care. Next slide, please. So the number of introductions affects the rate of takeoff. Yeah, but I want to.
Eric, is your internet on?
Was much less. If you imagine.
Eric,
sorry,
we missed you for a little bit, just as you started to explain this slide.
Okay.
It seemed like.
Can you hear me now? Sorry about that.
Yes, we're hearing you well now.
Okay. So I'm saying there are many things that affect the rate of takeoff of an epidemic in a country, and we won't cover them all. But 1,300 introductions in a place like the UK is the equivalent of going to a piece of grassland and starting 1,300 separate little fires. And in Kenya, we started with 10 little fires. So that's a huge difference in the number of little fires that start up before they have an opportunity to really start burning and moving through the rest of the grassland. And so that is one of the explanations why things have been much slower in many sub-Saharan African countries, except for South Africa, which is that very stark red line in this graph. So there are many things that affect this, but the number of introductions is a key one. And countries like Kenya did a really great job in putting in place fire control measures very early on. And we've seen the benefit of that with a very slow rate of increase. But that just re-emphasizes that now that we're basically allowing new grass to come in by moving around more, the risk of fires spreading is going to increase as we unlock a little bit. Okay, next slide, please. Okay, another big thing that's been a lot in the news is the issue of face masks. For many of us, actually, this global consternation around face masks seems a little strange. Those who live in Southeast Asia have been living with face masks for a long time, and very quickly, in certain parts of Kenya at least, and other sub-Saharan African countries, face masks have become quite normal quite quickly. But there is some global difficulty around the whole issue. Why? Well, the issue of face masks, whether face masks were useful or not, was based on what the World Health Organization knew about influenza viruses, which is that those viruses are transmitted through droplets and not through aerosols. And I have a couple of slides later to illustrate this, but I covered that in a previous presentation, the difference between a droplet and an aerosol. Anyway, aerosols are much smaller particles than droplets. Basically, when you cough, a droplet is a a piece of a small globule of saliva that exits your mouth and is quite heavy. Whereas an aerosol is something that can hang around in the air for much longer because it's much, much lighter. Okay, so because they are bigger, droplets are heavier and fall out of the air faster. And because the World Health Organization assumed that, like influenza, SARS-CoV-2 was transmitted through droplets and not aerosols, that was one of the reasons why face masks were not immediately recommended as a measure. And this is a quote from an article in Nature the other day saying, "For months, WHO has steadfastly pushed back against the idea that there is significant threat of coronavirus being transmitted by aerosols and that it can therefore accumulate in poorly ventilated venues and be carried on air currents. The agency has maintained that the virus is spread mainly through surfaces and bigger droplets that travel a shorter distance. But recent evidence and a recent push from a group of scientists you may have seen in the news, there were 200 or so, I think, people signed a declaration saying that this needed to be reconsidered based on recent evidence. And next slide, please. Next slide, please. Can you hear me? Yes. So the whole issue of droplets versus aerosols became very debated scientifically, and unfortunately, it's also become quite highly politicized in some areas. I won't cover that, but if you go and look at CNN, even this morning, you'll see there's all kinds of stuff around masks being of personal choice, and it all becomes wrapped up in a certain amount of politics in some countries. But nonetheless, this is a diagram that shows really what I've just been saying, that if you're a virus in a droplet, the droplet is heavy, and the distance that it can travel is of the order of 1.5 meters. If it is an aerosol, it can hang around in the air for much longer, which is why in some of the thinking around ILRI offices reopening, those offices in countries where there is air conditioning need to consider the risks of aerosolization much more than places like the Nairobi campus, where most people are ventilated with open air, as Andres Palau has been thinking a lot about this in the last few months. It's amazing how we can go down a rabbit hole of getting really interested in the way in which we ventilate our offices, because it matters in this context. Next slide, please. And on the next slide is really an image from the BBC, which I think explains some of this a little bit more clearly than the other image. Basically, droplets travel less far than airborne transmission. And again, that picture of someone coughing really shows those droplets become aerosolized and then can hang around in the air. Next slide, please. I think the next slide is for Dieter. Oh no, one more for me before Dieter takes for one slide. So in terms of face masks, the things to consider are how well you wear them and also why you're wearing them. You're wearing a face mask not because that piece of cloth over your face is going to protect you from someone else coughing, but it's going to protect those other people from you coughing if you're carrying the virus and you don't know about it. And if everybody's wearing a face mask, everybody is protecting everybody else. And I think perhaps that's one of the reasons why this has become a bit politicized, because you wear a face mask not to protect yourself, but to protect everybody else. And if you think only about yourself, then you don't need to wear a face mask. But in this situation, we all need to be thinking about everybody else around us all of the time. And this graphic that I pulled off Twitter on the right-hand side, I think shows it in a slightly rude way, perhaps. But if nobody wore trousers and you peed on someone, they would get wet. If they were wearing trousers, they would get wet less. And if both of you are wearing trousers, no one would get wet. And it's basically the same with a face mask. So think about peeing on other people being quite equivalent to a face mask. Apologies for using that slightly rude analogy, but I think it illustrates the point very well. And then the way you wear your face mask is also really important. And I've got some arrows here showing that a face mask that's pulled down over your chin, or that doesn't cover your nose, or that you're touching all the time is not going to be as effective as a face mask that you just leave alone, covering all the right places on your face. Next slide, please. Okay, yes, I'm really emphasizing on this issue because I think it's really important. So this is my last slide on face masks. And this relates to a study that was done for a group of people who were singing in a small choir in the US in March, showing that one individual had COVID-19 and came into that choir and sang with another group of people. And then there was a second meeting of the choir, as that graph on the left shows, a few days later. And as a result of that, one infected person who was standing in a crowd of people who, let's admit it, when you're singing, you're aerosolizing saliva all the time, they infected a very large group of people, 32 confirmed cases and 20 additional probable cases, and unfortunately 2 deaths resulting from this one infection event. So please take care in the supermarkets, in public transport, in church, in wherever you go where there are other people, to make sure that you're not participating in being that unknown case, spreading it to many others. Next slide. Okay, sorry, Dieter, your slide is coming in a little bit. I thought it was earlier on, so do be patient. So what about surfaces? This is the whole issue of surfaces and the need to clean surfaces has also become a little bit controversial, and it relates in part to that whole idea of whether the virus is in droplets or in aerosols. Of course, if it's in droplets and they fall out the air very quickly, it means that if you're sitting in a chair, the immediate 1 meter space around you could become very contaminated because you're shedding lots of droplets into that space. Well, of course, if it's aerosolized, it's spreading much further, but that also means that physical surfaces are perhaps less at risk. And this quote was published in the Lancet last week, I think, saying this individual's opinion, it was that the chance of transmission through inanimate surfaces is very small, and only in instances where an infected person coughs or sneezes on the surface and someone else then touches that surface within 1 to 2 hours after that cough or sneeze. So this doesn't mean that we shouldn't stop disinfecting surfaces and taking care with surfaces, but that the early worries about how important surface to person transmission was perhaps a little bit overdone, largely because the experiments, which are summarized in the graphic that I've put on this slide, used extremely high doses of virus that are much higher than you would actually be coughing out of you. And so it was a little bit overblown. That doesn't mean surfaces aren't dangerous. It doesn't mean we should stop cleaning things very carefully, but it does mean that person-to-person transmission is likely to be much more important than surface-to-person transmission. That's really the message I want to put here. And therefore, wearing your face mask becomes even more important. Okay, next slide. Right, this slide is for Dieter, so I'll mute myself and come back in just a moment. Dieter, over to you.
Okay, thank you. That was nice, and I think there were interesting questions in the chat. I think we need to come back to some of the questions later. You know, last time we had an update on vaccines by Vish, and we thought it's good to have an update on the therapeutics, because the therapeutics will be earlier on the market and more on the market than vaccines. But everything what we know now is mainly for therapeutics which are used for severely diseased COVID-19 patients. So the COVID-19 pandemic continues, and researchers and the manufacturers are moving potential therapeutics into clinical trials at a very fast pace. And the search is on to find treatment candidates that lower the mortality rates and reduce the severity of COVID-19. So this is the target of the development of the pharmaceuticals. And there, in contrast to last time, there are potential, there are now 3 therapeutics are approved to treat COVID-19. Approved in this case means approved on emergency approval regulations. So the one is dexamethasone, a generic glucocorticoid. It's approved normally for rheumatic and skin diseases, for allergies and asthma. And it got an approval in UK. And it has been selected as a possible therapy due to its potential for reducing the inflammation associated with cytokine release syndrome in patients with COVID-19. So it's clear it's not working against the virus. It's just only the cytokine release, which comes with a lot of inflammation, and there's information that many people just die on this reaction and not dying on the virus. So and the first preliminary results are promising. 35% of the reduction in mortality among patients on ventilators, and 20% reduction in mortality for patients receiving oxygen. And you see here again, these are severely diseased patients. Then there's a favilavir, is from Fujifilm Chemicals, and it's an antiviral agent, so it works against the virus, and it has approval in China and Italy to treat COVID-19. And there's a generic of this product which has an approval in Russia. So there are many currently clinical trials in phase 2 and 3 in China, Japan, and Canada. And the preliminary results say it's not effective in improving the clinical recovery rate of patients. It was not effective in treating mild or moderate cases of COVID-19. And interestingly, you know, why do countries like Canada approve a product which is not effective? And this is related to the regulatory authorities' different working style. Before, you know, there was always a risk-benefit balance, and this is now a benefit-risk balance. So in former times, the regulatory authorities looked first at the risk, and if the risk was low or zero, then they looked at the efficacy of a product. And under COVID situation, they changed it. And that's why it's possible that the benefit is checked first and the risk later. And so it can come to a situation that a product is approved, even if it's only emergency approval and is not working. The most promising one is remdesivir, an antiviral product from Gilead Sciences, and it's approved in Japan, but even FDA allows the use, and there's conditional approval in Singapore and in Europe, and it's also used as a conditionally approved product in the Arabic Emirates and in India. It was developed initially for Ebola and Marburg virus infections, and ultimately proved less effective than other therapeutics, but has shown effectiveness in animal studies against other coronavirus. That's why this test 2 phase in 3 trials are up in testing. It's an intravenous drug that inhibits viral replication, as shown in vitro and in vivo activity against SARS-CoV-2. So you see intravenous drug, which has to be given for 5 days. So again, only a product for severely diseased people and only use in hospitals. The preliminary results, it's beginning to show promise as a therapy that improves time to clinical improvement, but not an improvement in mortality. So results from over 1,000 patients in a study showed that remdesivir improved the time to clinical recovery from 15 days to a median of 11 days. So this is the benefit of this product, is that the disease time is reduced. But we have also to see it's a very expensive product. The typical 5-day treatment will cost about $2,500, and this is for government purchases from developed countries. And Gilead recently doubled its supply goal to cover more than 2 million patients by year end. So even here you see the limitation by price, limitation by the number of products being available. But there are ongoing negotiations with generic drug makers hoping to get the cost of this product down. So the next group are the anti-malarial, and there was one of the best promoters was Donald Trump, and he believed there's a scientist in south of France who believe it's a good product. And now we have met Bolsonaro, another promoter of this product, and he's taking hydroxychloroquine as treatment despite studies finding it is ineffective. He also shared a video of himself gulping down chloroquine, and what he said was his third dose already. And then in another video said, I trust hydroxychloroquine, and was smiling and said, are you trusting this product as well? But in the meantime, there's mounting evidence indicates that hydroxychloroquine and chloroquine, once a leading therapeutic candidate at the early phase, could cause more harm than benefit in patients with COVID-19. Particular side effects with heart failures are known, and the WHO and all the big organizations are warning for using this antimalarial product. Then we have the convalescent plasma, and I talked about this already, taking plasma from people who have had the disease, but were then okay again. And researchers have theorized that convalescent plasma could be used as a passive immunotherapy to help neutralize the virus. And the good thing is the preliminary results, safety data from 5,000 hospitalized adults indicated that convalescent plasma is safe for use. And there's a first peer-reviewed study of convalescent plasma, unfortunately only in 25 patients, but 19 of these patients, so it's 76%, with severe COVID-19 who received convalescent plasma had clinical improvement. In another clinical trial, mortality rate improved by 12%. Then there are another group, the therapeutics approved for other indications. I just mentioned two here. The one is ivermectin. I mentioned ivermectin because I was part of the group developing this endectocide product. Endectocide means it works against internal and external parasites, and it was developed for livestock. And then we realized that suddenly people took it against river blindness, so the onchocerciasis, and it was proved then finally, and the company Merck US is giving millions of doses to WHO to treat onchocercosis, so it's also known to have a very good safety profile in people. Ivermectin has proven effective only in vitro up to now, and it's inhibiting SARS-CoV-2 within 48 hours of treatment with a 5,000-fold reduction in virus. And now Japan plans to test ivermectin against COVID-19 in a clinical trial. And for all the golf players in the group, it was one of these surprises. A golf player, a scientist of Merck, took a sample from the green of the hole 18 and used this for feeding the mice, and the mice got rid of all the worms and mange. So it was one of the fantastic stories how products are developed just by chance and not by a good concept. Interleukin-6 receptor antagonists, several companies work on them, and they were developed for rheumatoid arthritis, and there are many of them on the market. There's one called Actrema from Roche, and this is tested now intensively, and the drug improves significantly clinical outcomes of pneumonia associated with COVID-19. So it's a 45% reduction in mortality. But, and that's the negative thing, other studies couldn't find any statistical evidence. So even that is not a promise really forever. So, and then we have the novel candidates. These are a lot of antivirals. very promising are the monoclonal antibodies, and there is also a recombinant fusion protein, which is also promising, but it's very much in the early research phase, so I'm not talking more about that. Thank you very much, and over to Eric again.
Thank you very much, Dieter. I was just looking at some of the messages in the chat about surfaces, and I just want to make sure I haven't created any confusion in what I said earlier. I'm not saying that surfaces are not a risky thing. It's just that the lifetime of the virus on a surface and the chances of acquiring the infection from a surface are lower than we previously thought, but that doesn't mean that the risk is zero. And certainly if someone with COVID coughs on their hand and then opens a door, that door handle could have virus on it, and we should still be very careful with those kinds of things. But the balance of evidence seems to show that airborne transmission is certainly more important than the surface. So that I just wanted to clarify that. Okay, I have a few slides left. I wanted to say something a little bit about antibodies because they continue to be in the news and in the literature. And this is the same slide that I've shown you before, looking at whether antibody tests could be used as some kind of immunity passport, allowing decisions on coming back to work, all the issues about protective immunity. Actually, the state of play is that things haven't changed enough for us to really update this in any substantive way. We still don't really understand what the immune response is, how long it lasts, and so on, remembering that this is still a very, very young epidemic. So next slide, please. So as a result of that, this is a report published by the Africa CDC just last month. And on the points that I'd raised about antibodies, they specifically say, at this point, antibody tests are not recommended as a criterion for discharging patients from hospitals, or indeed as a criteria for workers, including health workers, to return to work. So we're not yet at the place where we can use antibody testing as a decision, part of the decision about coming back to work. Next slide. Okay, this is my last slide. I just want to re-emphasize the three most important things that everybody, the three most important things that everybody can do. One, wash your hands regularly, as you have been. Please continue to do so. Second, wear a mask and wear it properly. And third, keep your physical distance as much as possible. And those three things are the best things that we can currently do with the technologies that we have to prevent the transmission of COVID-19. Thank you.